This recording is going to be about CDK proteins, or cyclin-dependent kinases. The reason why we're talking about these is because we've been talking about the cell cycle and mitosis, and CDKs are a crucial part of the cell cycle and mitosis. Here we see the cell cycle mapped out, and these stars are checkpoints, or places where the cell has to self-assess to determine whether or not it could enter into the next stage of the cell cycle. There are also checkpoints during mitosis, but those aren't shown in this image here. And the checkpoints are largely pertaining to the condition of DNA and whether it's been damaged or not, whether it's been duplicated, if it should be duplicated at that point after S phase, and if it's still duplicated in G2. And there are molecules that do all of this checking. Now, some of the molecules involved in these checkpoints are called CDKs, cyclin-dependent kinases. And these proteins are present in each of these different stages. So in other words, G2 has its own cyclin-dependent kinase. Mitosis has its own cyclin-dependent kinase. And each of these stages needs their own CDK because it's these specific CDKs that are going to signal to the cell that, hey, we need to move into the next stage. So in other words, when the CDK for mitosis is active, then the cell knows to go into mitosis. Then the CDK for G1 might become active, and the cell knows to leave mitosis and go into G1. So these are some cartoons of the CDK and the kinase, the CDK and the cyclin proteins. So CDK again stands for cyclin-dependent kinase up here. And so that is this red little Pac-Man looking thing. And then this other protein, the green one, is the cyclin. Now this protein, the CDK, is going to be inactive unless the cyclin is bound to it. And so these are also specific. These come in pairs. So there is a CDK for mitosis, and there is also a cyclin for mitosis. And the CDK for mitosis can only bind to the cyclin for mitosis. It can't bind to one of the other cyclins. It can't bind to the G1 cyclin and become active. So when we say that this protein is active, we mean that it's an enzyme, and its enzymatic action, or its job, is to actually add phosphate groups to other proteins. And that's kind of beyond what we've talked about so far, but essentially, to really simplify and almost oversimplify, when these CDKs are active, they're going to turn other proteins on or off. They're going to make other proteins active or inactive. And by doing that, they influence what's going on in the cell and help the cell to move forward into the next stage of the cell cycle. And so I compare this to a car where you can have a car present, but the car is not going to be active unless you have the key to start the car. And that's very much like how a CDK might be present, but it's not active unless you have the cyclin that can activate it. Now, I wanted to give this example, which is at the beginning of mitosis, or which brings the cell into mitosis. There is a third protein that we're going to introduce here. That protein is called cdk25 phosphatase. And this protein can be activated or inactivated as well. This protein itself can actually activate the CDK cyclin complex for mitosis. So this blue guy here, when it is active, it is going to activate the mCDK complex. So again, CDC25 phosphatase can turn on mCDK when it's bound to M-cyclin, and then this complex is going to become active, and this is going to interact with other proteins to either turn on their function or turn off their functions so that the cell can then move into mitosis. Interestingly, this is what we call a positive feedback loop, so that when this mCDK is active, it's going to activate more of these molecules indirectly because active mCDK activates CDC25 phosphatase. And so when it activates this blue protein, that blue protein can activate more mCDKs that were inactive previously, and they're going to activate more of the CDC25 phosphatase until you have the maximum number or every single mCDK active. And this is not the only protein that mCDK interacts with. It interacts with a lot of other proteins as well, but this is one instance where you could see that it's monitoring and modifying the action of other proteins in the cell.